All right, thank you so much for the introduction. Like you just mentioned, my name is Shari Rohret. I am a fourth-year PhD student. That seems crazy. And today I'm going to be talking about a recently funded NSF project that will focus on benthic meiofaunal diversity in cave systems within Cozumel Island and the Yucatan Peninsula mainland. So just as an overview, I'll briefly give some background about the organisms that we're interested in studying within these systems, and I'll spend the rest of the time talking about our plans for this NSF project within the next couple of years, and the analyses that we hope to and plan to conduct. And then at the end, I'll show some preliminary results of foraminiferal observations from some of these cave sediments. So given that Anchialine systems are the focus of this conference, I'm going to breeze past this, but I just wanted to mention, since I'm the first talk of this Mexico and Caribbean session, that the cave systems within the Yucatan Peninsula are extensive and anastomosing cave networks that we have the privilege of diving and sampling in. So again, most of you have conducted this research, we're the pioneers doing this research, but most of the research that has been conducted within these cave systems has been focused on pelagic crustaceans, many of which are endemic, and many of these have been morphological descriptions. More recently, there have been genetic analyses that are being conducted, and we're just starting to learn about ecosystem functioning within these cave systems and how macrofauna are interacting with bacteria. What we're trying to focus on with this NSF project is looking at the role of benthic meiofauna and their diversity patterns within Yucatan caves. So meiofauna are a size descriptor rather than a taxonomic description. So they include many groups, including nematodes, copepods, amphipods, etc. And also Foraminifera, which is what I am particularly interested in. So this term refers to the size of the organism, and changes in meiofaunal communities can be useful indicators of environmental change because they respond quickly to changing conditions, and they're also critical to marine food webs as they act. They're essential for transferring energy to higher trophic levels. So the meiofaunal communities that have been described in the Mediterranean, Atlantic, and Australia have been compared in many instances to deep sea fauna, likely because of the similar conditions that they are enduring. And meiofaunal communities in Yucatan have been limited, and the role that they play in ecosystem functioning is largely unknown. So within this broad umbrella term of meiofauna are Foraminifera, which are a group of meiofauna that are microbial eukaryotes. So Foraminifera are used often because they make these fossilizable tests. So they are very important in paleo-environmental and paleoclimate studies because the tests that they're creating can tell us valuable information about the environment in which they were living. So I could talk all day about forams and the really cool things that they can do, but I just wanted to mention something that you may not know, which is that although forams were traditionally thought to be strict aerobes, we are finding that they're capable of many different types of anaerobic metabolism, including complete denitrification and several other pathways. So in addition to that, they also have a lot of adaptations to low oxygen, including hypoxic and anoxic conditions. And so a lot of these are ultrastructural changes. So I'll talk a little bit about this later, but they're really resilient and they're able to survive very well in extreme conditions. So now that I've given a little bit of background, I want to talk about the overall goal of this project, which is to establish the diversity and ecology of benthic eukaryotes across environmental gradients within these cave systems, and also to determine how the benthic meiofauna are interacting with elemental cycles, and the role that they play in food web interactions. So to do this, we have a great team of divers and scientists who are working on this. We actually just got back from the first sampling trip on this project a little over a month ago, so we're really excited to be analyzing that data, and hopefully we'll have a lot of it analyzed before our next trip in February. So one of the things that we're doing is looking at spatial variability. So we're looking at 2 caves on the Yucatan mainland and 2 on Cozumel Island. In addition to that, we're looking at a transect within this cave, so we're going to be looking at how things are changing with increased distance from the cenote or sinkhole opening. In addition to that, we are looking at temporal variability. So to do that, we're sampling during wet and dry seasons. So we have 5 sampling trips that are funded under this project. We're going to be going about every 6 months. And we're also deploying what I think is a pretty impressive amount of long-term loggers, which will help us to look at changes in dissolved oxygen and conductivity within these systems. So the first aim of the project, and what I'll focus on here, is looking at the physical and chemical parameters that drive benthic eukaryotic diversity patterns. So to do this, one of the main things that we'll be doing is metabarcoding. So we're going to be looking at using general eukaryotic primers as well as foram-specific primers, because they have weird genomes and general primers do not work with them. In addition, something that's going to prove to be important is sequencing individuals. So we're going to sequence individuals, which will help us to build up a reference library for the species that are found within these systems. And then something that I will be working on is foraminiferal viability and distribution. So as I mentioned before, forams have been used a lot in paleo studies. Generally, this is looking at total assemblages, so that's both the living and dead composition of these foraminiferal assemblages. What I'll be using is a fluorogenic probe. So this is something that is incubated in bulk sediment samples, and it will help to identify only individuals that were metabolically active. So we can look at the composition of living foraminifera rather than total assemblages. And another thing, something that my advisor has been quite prolific in, looking at forams in deep sea sediments, is looking at their cellular ultrastructure. So this can tell us a lot about the adaptations that they employ to live in hypoxic and anoxic or any sort of extreme environment. So this is an image that I took from a foram from a hydrothermal seep, and some of the adaptations that they have are these complexes of peroxisomes and endoplasmic reticulum. They also often have a proliferation of lipids, so there are a lot of things that they're doing on the cellular level that helps them to survive and thrive within stressful conditions. So the second aim of this project is to determine the role of benthic eukaryotes within the ecosystem. So we're looking at ecosystem functioning. And these slides, we have a great team so we're going to be looking at stable isotopes of the meiofauna. I don't have time to go through all of the analyses that we're doing, but I just wanted to give you an idea of some of the methods that we'll be employing to look at ecosystem functioning within these cave systems. And then the third aim of this project is to establish the temporal constraints on benthic biodiversity patterns and ecosystem resilience. So to do this, we'll be using a lot of methods that I think we've seen previously. So we have both sensor-based and discrete sampling methods. For the sensor, we have the EXO that we take with us when we dive. We also have these long-term loggers that are deployed. And the discrete sampling, one of our co-PIs, he's doing a whole suite of chemical analyses that will help us to to get a better understanding of the environment as a whole, which will help when we are trying to look at these benthic diversity patterns and seeing what is actually driving changes. So in terms of preliminary results, so these are paired images. So these sets of 3 are the same specimen, just using different microscopy techniques. So these are some examples of the cell tracker using this epifluorescence microscope. So the bright orange areas, or they might look yellow on the screen, are where there is metabolically active cytoplasm. This is just a light microscope image, and the third image is an SEM image. So these are forams that were undeniably living at the time of collection. And what I'm seeing here is that there are higher abundances of certain taxa, such as miliolids, so these miliolid type taxa, and also these biserial taxa, including Bolivina. This is consistent with some of the paleo studies that have been done within this specific cave system. But one thing that's important to note is that we're seeing much lower abundances of living forams compared to when you're looking at the abundance numbers for the paleo studies. And another interesting observation is that often we're seeing that what is actually fluorescing is in the earliest formed chambers. So these middle chambers would be the ones that formed first. Here you can see these bottom chambers are the ones that are fluorescing the brightest. And this has been seen previously as a stress response for forams. Essentially, as things get stressful, they start retreating their cytoplasm into the earliest, into their first chambers. So that is interesting. Another thing that I've observed in a cave in Cozumel, so this is the species that I think are the same as these based on the elongated aperture and also this flared apertural opening. But as you can see, these forams are doing something very strange when it comes to building their test. This is likely a stress response. Something is happening chemically that is causing them to have these more free-form test morphologies. I saw a foram on our trip a month ago that had 2 apertures, which is really unusual for forams. So this is just an observation of these mutant specimens that have all sorts of weird morphologies. In addition, these were some rose Bengal stain specimens from a cave in the Yucatan mainland, seeing a higher abundance of these biserial types of forams, but there is a surprising diversity of these living forams that are in these caves. So something that I hope to talk about more today were the metabarcoding analyses, but we're still working on interpreting the results that we got back. So we're looking at meiofauna, including forams, so we're looking at bulk sediment, and we're using general eukaryotic and foram primers. Some of the challenges that we're encountering, which we'll talk more about if you go to the metabarcoding workshop tomorrow, low abundances of meiofauna, which in the bulk sediment is proving to be an issue. Also, something else is a lack of reference library, which is something that we're hoping to build up throughout this project. And then there are several other problems that I'm sure we'll talk about tomorrow. So in summary, this project is working to expand our understanding of community biodiversity and ecosystem processes within the Yucatan mainland and Cozumel caves. We're using a variety of interdisciplinary approaches, and these results will benefit aquatic benthic ecology and biology, biogeochemistry, and will also have implications for paleo interpretations. So with that, there's a whole team of people who are helping to make this happen. NSF is obviously our main funding source. Yeah, thank you.